tarde a todos. Estamos dando início a mais uma palestra de encontro universitário. Eu queria, antes de abrir a apresentação do palestrante, agradecer, em nome da Comissão Científica, a professora Jeane Matos, pelo convite. É uma honra participar aqui desse encontro universitário tão historicamente conhecido, né? Eu queria passar à frente e começar, então, a apresentação do palestrante, que é também uma grande honra fazer essa apresentação por conta do histórico da colaboração que nós temos com a Universidade de Virgínia, né? Então, o palestrante de agora vai ser o Dr. Richard Guerrant. Ele hoje trabalha na instituição Universidade de Virgínia, né? No Departamento de Doenças Infecciosas e Saúde Internacional, um centro de saúde global tão importante nos dias de hoje. O Dr. Guerrant é médico formado pela Universidade de Virgínia, ali na década de 68. Ele tem uma carreira profissional legendária para os colegas, inclusive, da Universidade de Virgínia. E ele tem uma formação em residência em doenças infecciosas e é o fundador do Centro de Saúde Global da Universidade de Virgínia, onde continua sendo diretor de fundação desse departamento. O professor Guerrant, ele tem uma vida profissional e participar de várias sociedades importantes na área de saúde. Ele é membro honorário e ex-presidente da Sociedade Americana de Medicina Tropical e Higiene. Também faz parte como membro da Sociedade Royal Society de Medicina Tropical e Higiene da Inglaterra, né? Ele tem várias outras participações em sociedades que a gente não vai ter tempo para comentar aqui. Mas gostaria de chamar a atenção do palestrante para algumas contas de publicações que o palestrante tem. Como eu disse, o professor Guerrant é legendário, ele tem mais de 70 publicações de artigos em revistas de alto impacto, Lancet. Nós temos aí 9 patentes, 8 livros publicados. O mais recente, Evolution of Evolution, do qual ele vai falar um pouco aí nessa palestra de 43 minutos. Bom, como recurso humano, chama a atenção, o professor Guerrant, como eu disse, é legendário. Só no programa, 45 anos, em colaboração com a Universidade Federal do Ceará, ele tem mais de 100 estudantes e técnicos que ele formou nessa colaboração. Muitos deles, obviamente, foram de iniciação científica também, e hoje têm mestrado e doutorado, e hoje ocupam cargos extremamente importantes em instituições, principalmente acadêmicas, não só nacionais aqui no Brasil, mas também muitos internacionais também formados. Queria só conotar que nós estamos 45 anos de colaboração internacional. O programa da Universidade Federal do Ceará e da Universidade de Virgínia de colaboração científica do CAPSPLIT hoje, ele é um programa mais longevo e ativo que você tem existente aqui na Universidade de Virgínia, e nós comentamos ainda ontem que nós não temos nenhum histórico de um programa tão longevo e ativo da Universidade Federal do Ceará e por fim, eu queria só comentar que o professor Guerrant, ele é conhecido por ter sido instrutor de vários colaboradores de nobelistas. Eu citaria apenas um deles, o Barry Marshall, que foi pós-Dr. Fellow com ele durante 2 anos, né? E em 2005 ele ganhou o Prêmio Nobel de Biologia ou Medicina, né? E é uma honra muito grande ter o professor Guerrant nessa palestra, né? Eu queria, então, só citar o título da palestra, está em inglês, mas traduzido, nós podemos dizer que ele vai enfocar os principais patógenos estão associados com enteropatia ambiental e infecção entérica, e mostrar modelos de pesquisas que têm sido construídos ao longo desses 45 anos de colaboração internacional. Então, é com prazer que nós damos início à palestra do Guerrant.
Pause share. Resume share.
Have your slide on.
Yes, but I don't see it. Okay, recording is in progress, I guess.
Yes.
All right. So, Aldo, I thank you and everyone there at the Universidade Federal do Ceará, our colleagues in the field work, because you and Reinaldo and everyone there have made it possible to bridge clinical and laboratory research. And so I will address how that bridge makes it possible for us to model enteropathy or diarrhea with all of the top pathogens in children in areas where it is a serious problem. And I especially appreciate all of our amazing collaborations over now in our 45th year, as well as Reinaldo and many others that are in my next slide. And that is showing here the amazing group of people that started on July 21st in 1977. Happened to be my birthday. I didn't realize until I dug these slides and dates out. But Dr. Ed Hook, shown here, and I visited Dr. Galba Araújo and Dr. Auxiliadora de Souza and Nogueira Paes and Manassés, and launched the collaboration that is now one of the longest international collaborations in medical science that I'm aware of. And it is the amazing people that over the years have made this such an incredible, productive time. Obviously, Anastasio and you and Noelia were our first fellows to come here, then Reinaldo, Gerli, and then we collaborated with Selma and with Pascal Bessong and with Edgar Carvalho's group in Salvador, and worked with your amazing team of Sayonara and the remarkable group of people that include Lucia, Hosanna, and Fatima, as well as people in your laboratory like Claudia, and then many students now in faculties around the world like Orleancio, Noronha, several wonderful Brunos, including your remarkable daughter, and many, many more. Most recently, old Pedro down here, Medeiros, who's written several papers. And here now is a remarkable young lady, Desiane Costa, who is doing great work. And you can see from these pictures that these collaborations with you have made great friendships that have lasted over all of these years. And because others may not, they all know you and Noelia, they may not know that this is my wife, Nancy. And we have done great things together that are fun as well as work. So this next slide simply shows some of the remarkable people that this collaboration has involved. You and I were able to get this crazy guy, Barry Marshall, to Fortaleza, along with Barry Farrer, and meeting in your lab to talk about H. pylori infections. And indeed, Barry won the Nobel Prize a few years later for having discovered Helicobacter pylori as the cause of peptic ulcer disease, as well as of gastric cancer and lymphoma, and possibly even other problems like pernicious anemia. We've been privileged over the years because that connection with Barry led to Nancy and me having time with the person who shared Barry's Nobel Prize with him, the elderly pathologist, Robin Warren, who also won the Nobel Prize. This happened to be in Belfast, Ireland. We also have had people like Farid Murad at our lab meeting. And there are great people from your laboratory. Bless him, old Jonas, who's sadly no longer with us, Hosa, Renato Lohani, with Farid Murad, who won the Nobel Prize for finding G proteins and the activation by guanylate cyclase instead of adenylate cyclase. Al Gilman got another Nobel Prize before that with the discovery of G proteins. And then this, of course, is Dr. Professor Mohammed Yunus, who won the Nobel Prize, the Dalai Lama, and actually your president, Fernando Enrique, was visiting us here in Charlottesville. But the most important people to us have been here and there in the laboratory, like David Bolick, who is responsible for most of the work that I will share later. People like Lorna Bori, who are just amazing special friends who we've now departed. But Lorna was one of the amazing glues that held us all together. People like Jonathan Swan and Dick Pearson, Shirley Warren. And there you and Noelia are with, I think that's Selma and Anastasio, Jim Hughes, and many amazing people. Here's Leah Barrett, who was with Charlotte in this picture at our TropMed meetings. So with that as background, let me summarize the 4 main points that I want to emphasize in this presentation. And that is the possibility that we may have 3 new diseases that we need to recognize with early childhood intestinal infections. And that constitutes, in addition to diarrhea, a triple burden. The first is the decrement in growth that stunt children in their first 2 years of life, and they never recover. And we call that HAZDROP because nobody has ever paid attention to that. And it's as real or more important even than diarrhea itself. The second new disease that enteric infections probably do contribute to in a major way are the cognitive development of children that is robbed, that is taken away by intestinal infections in their early childhood. especially in the favela or shantytown settings. We call that a cognitive hit or cog hit. In fact, that can be as much as 10 IQ points with an Alzheimer-like deficit, to which I'll return because Aldo and Reinaldo have both done pioneering work in this area, because the Alzheimer's gene may be in the human gene pool because it protects those children under intense pressure. We can even mention how evolution itself is changing, the title of a little book that I have written recently. And finally, enteric infections may increase adult obesity, metabolic syndrome, or cardiovascular disease. Enteric infections in early childhood then leading to metabolic syndrome later in life. The fourth take-home point is the laboratory point. The other three are all clinical. And the fourth point is that promising models in the laboratory and genetic and metabolomic tools can help dissect the mechanisms and develop biomarkers and interventions for these terrible problems. Now, much of what I will present is down here in the bottom, and Aldo has the original slides because mine are not showing on the screen, covered up by the program here. But the reference for virtually all of the slides that I will show now is in here, and Aldo can share that with anyone who's interested. So if we look at the global burden of intestinal infections in early childhood, we realize that it is comprised of mortality, or the years of life lost, and the morbidity, years lost to disability. And you put those together and have the disability adjusted life years, which is a profoundly important public health point for all of health and disease and research. And the good news is that in the 45 years that we have been working with you in Fortaleza, there has been a dramatic decline in the mortality in the less than 1 year children, or 1 to 4, or 0 to 5 year old or 4 year old children over decades of time since then. However, the bad news is that the morbidity, this case it's diarrhea, but stunting, the has-dropped, cog hit, and metabolic syndrome has not declined. And a few years ago, it was still very high, if anything even higher with diarrhea. That has started to come down now. But if we pay attention to malnutrition, which is the leading risk factor for health loss, then we find that fully 140 to 160 million children are stunted, and that is moderate to severely stunted with a height for age z-score of less than -2. That represents 20% of all the children in the world and nearly a third of children in developing regions of the world. So how does intestinal inflammation stunt linear growth? This was frankly very confusing to me until we examined Aldo's data from his case control studies at IPREDI, in which he found that fever or cough or diarrhea or even fecal myeloperoxidase, representing intestinal inflammation, can each independently and highly significantly contribute to systemic inflammation measured by the high sensitivity CRP. That in turn is associated with a failure of the liver to respond to growth hormone and produce IGF-BP3, IGF-1, the mediators of growth. And it's because those mediators are impaired by these problems that, in fact, you lose the negative feedback, and it's confusing and counterintuitive that growth hormone levels are actually increased with stunted growth due to inflammation. And these references, again, are shown over here, and I don't know how to move this bar to let you see them without messing up the presentation. But Aldo has all of these references. And so I want to show the most amazing multi-center studies done at 7 African sites with over 9,000 children, and then at 8 sites in Asia, Africa, and South America, which is the Mal-ED study of over 2,000 children at these 8 sites that Aldo led the Fortaleza site of this study. And these studies both examined the causes of diarrhea, which have now been done by Taq polymerase with quantitative PCR, which is totally changing our view of enteric pathogens. But the viruses, bacteria, and parasites are obviously important for diarrhea. However, the Mal-ED study examined growth over an extended period of time. And there we find pathogens that are associated with growth failure or has drop that are different from the ones that cause diarrhea. Most of the viruses drop out, and so it is Shigella, Campylobacter, enteroaggregative E. coli, and the different types of pathogenic E. coli, as well as the parasites, Giardia and Cryptosporidium, that are associated with impaired growth. And it's a little different from those associated with diarrhea. Thirdly, the enteric pathogen burden and brand new data now emerging that Shigella itself and fecal inflammation, even without overt diarrhea, can associate with impaired cognition in these children in later years, after 4 to 7 years, which is exactly what Aldo and Reinaldo and we found in the studies there in Fortaleza in the 1990s that were published in 1999. Finally, the exciting news is that we can model either diarrhea or growth shortfalls with all of the leading pathogens for growth and many that cause diarrhea. Finally, we can even now begin to see evidence from both the field studies in children that have grown up and even in the models that diarrhea or modified gut function or inflammation can lead to metabolic changes that reflect metabolic syndrome in later life. So the question is, how did these infections contribute to inflammation? And this is an amazing, previously unpublished analysis of over 20,000 stool specimens. Maybe I can move that, or maybe I can't. Sorry, yes, okay. Over 20,000 stool specimens that were totally asymptomatic or silent, okay, no diarrhea. in these studies across Asia, Africa, and South America, including Fortaleza studies. And leading the list is a highly significant association of asymptomatic Shigella infections with intestinal inflammation. This is a dramatic increase. Excuse me. Followed by Campylobacter enteroaggregative and enteropathogenic E. coli and enterotoxigenic E. coli. Yes, group 2 norovirus slips in here. I'm quite sure that we do not understand that exception, but these are the leading pathogens associated with intestinal inflammation. Interestingly, I can tell you that unpublished data that are in preparation now suggest that Shigella and fecal MPO may also be associated across all the sites with cognitive deficiencies as well. And here are the viruses. Giardia actually is in the opposite direction, unless it is co-infection, and that it may be made worse. So that means that we may have a triple burden of these silent enteric infections without even diarrhea. And they could be either direct, like the direct effects on stunting or cognition or metabolic syndrome, or indirect, with stunting being also affected like cognitive impairment, or even obesity and early stunting. So each of these is a reference that is given in that original reference and then the papers that are cited below in this Nature review. Next, then, we looked back at these absolutely precious children who had been a part of our studies in Gonzalez Diaz like 7 to 9 years earlier. And we went back, and thanks to Aldo's long-term follow-up, we were able to do cognitive testing and discovered that the diarrhea burden, which is this curve here, sorry that doesn't show, but this is diarrhea burden. The higher the diarrhea burden, the lower the IQ. And there was an average of a 10-point IQ drop with the average burden of diarrhea, which is just a bombshell, is amazing. And then Reinaldo started looking at the ApoE allele frequencies, because the cognitive deficit that was most affected was similar to that seen in Alzheimer's disease. It was semantic fluency, not so much phonetic fluency, retrieval from another brain region. And that's when Reinaldo found that ApoE was associated, but instead of being bad, this terrible gene risk for Alzheimer's was actually protective of the children's cognitive development only when they had heavy diarrhea burdens, as if this is a thrift allele, the allele that lets this gene have an advantage when all of our ancestors, no matter where we are in the world today, lived through multiple bouts of heavy intestinal infections. And that may explain why this terrible gene now exists in our human gene pool and has risked Alzheimer's disease. This, in fact, was further shown when Orleancio and Reinaldo and we were able to get some ApoE4 targeted transgenic mice to show that those E4 knock-in mice actually have better growth, shown here, and better villus encrypt architecture and shed less cryptosporidium. So confirming in an animal model this hypothesis that ApoE4 can be protective against some enteric infections. An extraordinary, I think, finding that the world probably has not appreciated sufficiently, because that is something that if we got rid of diarrhea, then there would be no advantage to having this bad gene. It would take a few more millennia for that to disappear, but suffice to say, this has much greater importance than I think we've realized. In fact, it's remarkable how important those first 2 years of human life can be. That is when we, Homo sapiens, develop most of our synapses. We start at birth with very few synapses, and yet in our brains. But by age 2, we have peaked. And yes, we gain a few after that, but we trim more. So the peak synaptic density in the human brain is, for everyone on this call over 2 years old, has been a peak, and we're all downhill after that. And that's that early period when those synaptic tracks are laid down like a train track. It's also when these children are most vulnerable to that damage of synaptogenesis. Indeed, Reinaldo and colleagues were able to beautifully show that malnutrition indeed did impair synaptogenesis and cellular density in the hippocampus of the mice. This obviously cannot be done in children unless some scans someday may make it feasible. But this is clear in the animal model. And in fact, that synaptogenesis, which is shown here, is in fact impaired by malnutrition and improved by glutamine or zinc or vitamin A. And sure enough, back to the field, we go from the model to the field. We went from the field to the model, animal model, now back to the children. And indeed, Aldo was able to show significant improvement in some of the verbal learning scores in the Gonsalves-Diaz children. However, when they received a combination of glutamine and then zinc and vitamin A over a year, and most of that improvement was actually in the little girls, the females. And so that again shows the bridge between the laboratory and the field, which is so important to improving human health. Certainly is truer now today with COVID Omicron than ever before, perhaps, but it's very important. So let me turn to the models and just summarize that when we examine the mouse models of either enteropathy, which is defined in our model by weight decrements or growth failure, or diarrhea, defined by loose stools and along with the weight loss. And here we find that there are 3 critical determinants of the outcome. The first is the microbiome, which is altered by antibiotics. And it's fascinating that these are different with the bacteria versus the parasites. The second variable that's critical is diet. And we have examined in some detail protein and zinc effects on these different pathogens. And the third critical variable is the pathogen itself. So the parasites and the bacteria are all different, and they respond differently to these other two variables, the microbiome and the diet. For example, giving antibiotics really does not make Cryptosporidium or Giardia infections any worse. It actually makes Giardia infections even better. But without giving antibiotics beforehand, we do not get good bacterial colonization and outcomes of interest. Conversely, the protein in the diet is very important for Cryptosporidium, also to some extent for Giardia, but less important for the bacteria than zinc, which is profoundly important for the bacteria, as shown here. So let me take you through a couple of examples. And these are data from Bruno Coutinho's early work on a Cryptosporidium model. Let me try to move this mugshot here to get it out of your way. And here it is very clear that Cryptosporidium infection in the mice both causes malnutrition and worsens prior malnutrition. So it's a cause and an effect of malnutrition. Shown here is the nourished mouse growth by weight over days, many days of study. And if they get a Cryptosporidium infection, there's a highly significant 14% decrement in their growth. The malnourished mice grow less well, but when they get infected, there's an even greater decrement. And you add these together and it's devastating. This is crystal clear in the histopathology. Normal histology in the upper left histology panel, effects of malnutrition or of infection by themselves in these two panels, but on the lower right is malnutrition with infection, which is devastating in the mice and in the children. In fact, you can take the identical infectious dose of Cryptosporidium and give it to mice that are either nourished in the blue bars or malnourished in the green bars. And this is not subtle. This is a 5 log or greater increase in the shedding of Cryptosporidium parasites if you're malnourished with the identical infectious dose a week, 5 days before. That is probably because the protein deficiency impairs the cell turnover. But we can also look at both the microbiome and the metabolome and find this very complex analysis of all 3 variables in a 3-dimensional graph as shown here to show that the metabolite, which is fascinating but increased in Cryptosporidium infections, is not from the microbes, but is instead from the host, driven by the Cryptosporidium infection. The reason I mentioned that is because there is a clear correlation of trimethylamine oxide, TMAO, with heart attack, strokes, and death. It is, in fact, a better predictor than cholesterol. When we eat our phosphatidylcholine in dietary carnitine or lecithin in red meat or egg yolks, we obviously have that converted by gut bacteria into choline, which is then further converted to trimethylamine. And then under hepatic flavin monooxygenases, the conversion to trimethylamine oxide occurs. Well, malnutrition specifically affects trimethylamine. The ApoE knockouts lack these hepatic enzymes, and cryptosporidium infection enhances TMAO. So you can begin to see how specific infections, in this case cryptosporidium, can increase the risk of heart attack, stroke, and ultimately death from heart attacks or strokes or metabolic syndrome later in life, which we think we can see evidence for in our children and in the mice. So turning to the enterotoxigenic E. coli model, just as a quick example, and all of this is published in the overview review in April of this year in American Chemical Society Infectious Diseases, ACSID, that's down behind this bar here. If I can find a way to move it, but I cannot. Here's the effect of enterotoxigenic E. coli, LT plus ST, with house chow in the brown lines, and this is the infection and the control, and in either protein or zinc deficient mice. And you can see that protein and zinc deficiency has an effect on growth, but it doesn't make the ETEC infection any worse. In fact, the shedding is, if anything, slightly more in the house chow mice with this pathogen. That's going to be different with others. And the fecal biomarkers similarly show differences, but in this case, it's zinc deficiency that makes those much greater, whether it's fecal myeloperoxidase reflecting inflammation or lipocalin-2 reflecting both either inflammation or intestinal damage. So let me return to that slide that showed that Shigella was #1 at causing intestinal inflammation in the children. So a Shigella model was very important. Here again, we see the biggest difference is with the house chow mice. They actually get diarrhea and recover. So the infection in house chow mice is actually, however, able to be reduced over time, whereas zinc deficiency this time actually prolongs and worsens the infection, and that becomes even clearer with the fecal inflammation. So the message from the mouse model suggests that indeed the infection in zinc deficient children may be even worse or have a greater impact. In fact, we can use these models for the demonstration of vaccines, like in this case, the combined vaccine of Shigella and ETEC that was done in collaboration with Dr. Eileen Berry, and it was Pedro Medeiros who put these data together, and they're in the hidden references down here. But suffice to say that it is fantastically powerful for us to be able to show in the same model that the same vaccine that is a bivalent vaccine against Shigella or ETEC can have beneficial effects on either preventing Shigella or ETEC infection outcomes. Furthermore, we can show that there's a new vaccine that suggests that Shigella-induced weight loss can be prevented. So the models are tremendously powerful at helping us select the best vaccines that are likely to have effects on growth or diarrhea or even cognitive and other outcomes. These are the effects on Campylobacter infection, for example. And here, zinc deficiency becomes profoundly important because it's only with zinc deficiency that we get severe infections in terms of weight loss or even colonization beyond a week or so. And that's evident with even bloody diarrhea in the zinc deficient mice, which Campylobacter we know can cause in children. So that can be shown histologically as well as with fecal biomarkers or cecal contents that persist in day 2 at peak, but persist even out to day 14, which is after much of the infection may be resolving. And that enables us to test pilot candidate Campylobacter vaccines like this one that Christine Zemansky and Harold Malthaft have developed. And so this becomes a very powerful way to examine multiple pathogens and interventions or vaccines that can make a difference. So let me conclude then with the vicious cycles of enteric infections with or without overt diarrhea that can cause malnutrition or even cognitive impairment or vaccine failure that in turn become a vicious cycle through impaired immunity or worsened infections with diarrhea or infections themselves. In fact, if we start examining the interventions, every one can become critical to preventing this vicious cycle, and each may be insufficient alone. And we likely will need multiple points of attack at either antimicrobials, vaccines, repair nutrients, a special interest of Aldo's and mine, microbiota, probiotics, or obviously improved water and sanitation to reduce the infectious doses. That might not only improve the enteric infections, malnutrition, and cognitive impairment, but even some of the costly long-term effects, which taken together become vicious cycles with poverty itself. So my last slide returns to my introduction, where there are 4 take-home points that are most importantly, 3 are clinical, from clinical studies in the field, like Aldo has coordinated there in Fortaleza, that are so important. They are the effect on growth or HESDROP, cognition, COG-HIT, or metabolic syndrome. In fact, though the promising models can give us clues for how to dissect these interventions. Along the way, I can just mention that the little book on evolution of evolution that the ApoE4 protection illustrates is only one of many changes. Sickle cell anemia, for example, is one of those once advantageous when malaria was endemic that became disadvantageous when you survive and suffer the ravages of sickle cell anemia. And there are other traits that I mentioned in this little book on evolution of evolution, the survival value of caring itself, which we need to learn, especially now amidst the COVID crises around the world that aren't going away. In conclusion, then, the reason that we do all of this work is because of these children. And with that, I'll close and thank you and Aldo, Reinaldo, all of our great friends and colleagues there that have become, in fact, wonderful, lasting friends, as well as amazing collaborators. So with that, I thank you and will open for any time that's left for questions. Thank you.
Bom, yes, Dick, I'd like to acknowledge your well-packaged 45 years excellent research and findings. It's very interesting to see the consistent of the data between the field and laboratory, and then back to field. I think translational medicine is all over around this talk. That's interesting. I think there is several interesting people here around. Maybe they have some question. Now we can open for question if you will. But before they do, let me request Reinaldo here to have a question for you quickly. Yeah.
Hi, Dr. Guerrant. Congratulations for the great talk. Very nice overview of your research and the outstanding collaboration between the Federal University of Ceará and the University of Virginia. My question is whether you still keep the stools from the different infection models you have, so you may kind of do an infection to other animals like APOE knockout mice to see whether these microbiota from these animal models, infected mice, could increase cardiovascular disease, and whether TMAO may be involved. So I would like to hear your thoughts about that.
Reinaldo, muito obrigado. As always, your questions are incredibly insightful and important. We are limited, Reinaldo, in terms of the amount of specimens that we can continue to save year after year. And we have encountered limitations that are really twofold. One is samples over 45 or more years are a problem to keep at -80 for forever. And so we do not have all of those anymore. But you ask also about the samples from the mice. And the problem there is that we only get minuscule amounts that David Bullock has succeeded in doing amazing gene probes and all sorts of cytokine and protein assays. And so most of those have been used up. Aldo and I have both worked at trying to save as many of the samples from the children as possible. And we've, in fact, already gone back several years to retest some of those for work that has been published, I think in PLOS ONE, among others. But to adequately answer your question about does a specific infection lead to metabolic changes like TMAO that would make a difference from the models, we've published our Cryptosporidium data on that, which I actually showed, but that would require, I think, another set of experiments to get adequate samples that are designed to get what you want. If you want peak infection, then, sacking at 3 to 5 days might make more sense. If, on the other hand, you want the persistent infection, then you develop the model with different diets and pick a different time to try to get more material for assay. Does that address your question? I'm sorry I don't have an easy answer that we have everything, but we really don't.
Okay. Thank you, Dick. Thank you.
Great.
Dick, while there is still coming some question, let me maybe comment and maybe say your opinion on this thought. I mean, right now we try to investigate metagenomic of the stools to see not only pathogens, but also maybe microbiome association with pathogen, and I think it's going to be an enormous, question important to associate this with enteropathy or maybe malnutrition or cognitive or growth and physical activity or education. How do you see this in terms of perspective to pursue some work? It's a very intriguing thing.
Well, Aldo, you also are amazing at raising critically important points, and I could not agree more that the microbiome is clearly playing an important role. The best and clearest example of that is the data that we clearly have showing that without prior antibiotics, the bacterial infections are very difficult to get in the mice. And then we stop the antibiotics a day before the infection, but that obviously is a microbiome point to which you allude. The role of fecal samples, though, in assessing not only the microbiome, but even a lot of the human genetic material is just beginning to evolve with technology that is frankly amazing. And as you know, with respect to genetics, we have salivary DNA from a lot of the children. And Alex in your lab has worked quite a lot with that. In fact, it's been a struggle to get that finished before those samples get discarded. They are even more precious in many ways because it's all just the human genome that has already been analyzed for genome-wide association studies, GWAS. And yet we haven't adequately examined that. So I think the capabilities are burgeoning, are increasing dramatically, and the limitations are really on our ability to pay attention and use this growing huge amount of information. But you're absolutely correct. That's an important gold mine that we need to be understanding and studying better. Yes, thank you.
Yeah, wow, thank you. That's huge important, no question. Let me see if there is any other possible question around. I think no. And I would like to close this conference to acknowledge again you, Jake, for your disposition for this talk. And it's not easy, we know, but the package is saved. So and also it's good to overview all the time because there is a lot to think and to translate to the community, as you mentioned. And thank you very much for this talk.
Thank you, and everybody there at UFC, and muito obrigado. Thanks.
See-
Ciao.